This conference will now be recorded. This conference will now be recorded.
I'm audible. Good evening, Millie. Am I audible to everyone?
Yes, ma'am. Yes, ma'am.
Okay, fine. Okay, so good evening, everyone, and hope you're enjoying the classes. And if you have missed yesterday's lecture of Indian Sir, you can watch it on YouTube. And please register with us so that you can what you want. Fine. So today's our topic is nucleus and transport mechanisms. Here our main target will be how the proteins are transported in and out of the nucleus, and some important points of the nucleus which comes in your net exams. Okay, now as you can see in your first slide, here I have put a cartoon and a fluorescent image. Okay, so this is a cell and he is accusing and he is making fun of this organelle. This organelle is nucleus. Okay, this is nucleus. And here you can see a word called as DAPI. Okay, now from this figure, your 1 4-mark question will be solved, which comes in your analytical part, that is part C. And it is a research-based question in which you will be given an animal cell culture and you will be asked to find out that how you will recognize a nucleus in an animal cell culture, whether a protein is present or not in a cell, and how you will identify that. The answer is your DAPI fluorescent label. Okay, this is a kind of staining which will stain your nucleus. DAPI is specific for nucleus and you can see the color is blue. Okay, these all things which you can see here are the nucleus. Okay, and only the nucleus will be staining with the DAPI stain. And why this DAPI can stain only this nucleus? Because DAPI has affinity to bind with the AT rich regions. What are AT rich regions? What are AT? Have you heard about ATGC? Anyone? Have you heard about ATGC? What are these? Yes. What are these? ATGC? Nucleotides. Yes, very good. They are nucleotides. So these DAPI, they will specifically go and bind to these AT rich regions, and that is why your nucleus will be stained blue in color. It will be blue in color, which you can easily see in your fluorescence microscope. So from here, your 2 marks question and a 4-mark questions is solved. And in your analytical types of question, one question you will definitely find on nucleus. And here it is, the answer is DAPI. You have to stain the organelle with a specific fluorescent stain, and that is DAPI. Okay, so here the nucleus is very happy and he's telling at least I can be noticed. Okay, notice means at least it can be seen in the full cell. If you are making it stain with this DAPI, it is going to be visualized. Fine. Okay, so this is something about myself. And if you have any query, you can email me on this. I get mails on this and I try to answer the queries also. So if you have any doubt related to any competitive exams, you can mail me on this email ID. Fine. Okay, so this nucleus, it is called as the information center of the cell. Why it is called as an information center of the cell? Now see, we have nicknames for many of the things. Okay, my school name is different, and my parents may be calling me with some nickname. So like everything has some nickname if it has special characteristic. So here our cell organelles also, they have important nicknames. Like nucleus, it is called as the information center of the cell. Okay, information center of the cell. DNA is the blueprint of the cell. Mitochondria are the powerhouses of the cell. Lysozymes are the suicidal bags of the cell. So these are the nicknames which are specifically targeting, which are specifically mentioned. The names are mentioned specifically targeting their functions. Okay. So the nucleus is called as the information center of the cell. Now you can see here that the nucleus is a double membrane structure. It is a double membrane structure. You can see it has having two membranes here. The first one is, this is your first one, which is called as the outer nuclear envelope. This, the outer one is called as the outer nuclear envelope, and the inner one is called as the inner nuclear membrane. Okay, so our nucleus is a double membrane structure having two layers. The first is the outer nuclear envelope, and the inner one is called as the inner nuclear membrane. Now both of these are very, very important. We will see in the consecutive slides, but both of these are very, very important. Now our nucleus is not closed body. Okay, it is not a closed body, it is an open body, and this membrane is a selective permeable membrane. It is a selective semi-permeable membrane. Okay, why it is selective? Because it will not allow anything to enter inside the nucleus. It will not allow anything, means it has to be regulated. The things which are transferred in and outside are regulated, just like the borders on our country. Anyone cannot enter in our country, or anyone cannot go out of the country just like that. Anyone cannot enter our country just like that, because they have to be given some specific gate passes, or like that, passport, visa, or something like that. The same thing is here with our nucleus also. And why this is and how this is regulated, you can see here, there are something called as the nuclear pores. These are called as the nuclear pores. Okay, these nuclear pores are very, very, very important, because you can see these are bouquet like structure, like flower like structure. Okay, and these are the pores which will regulate the entry and exit of all the cargoes. Cargoes means anything, whatever is coming inside or whatever is going outside the nucleus. So these are the things, the nuclear pores, this will regulate everything. These are called as the nuclear pores. Very, very important. These pores are present over the nucleus. These are present over the nucleus, and we will see in the next slides, these nuclear pores are continuous with this. These outer and inner nuclear membranes are continuous here. We will see in the next slide. Okay. Now see, this is our DNA. The main, what is the function of this nucleus? The function is it stores the genetic information. It stores the DNA. That is why it is called as the information center. Why it is called as the information center? Because it has our DNA. It has our genetic material. The heterochromatin and the chromosome territory. You don't have to worry about these words. Just remember that there is DNA which is present inside our nucleus. And you can see there is something called as the nucleolus. It is very important, and you can see from the figure also, and this is the 2 marks question which is asked in your B part, that which is the largest organelle which is present in the nucleus. Which is the largest organelle which is present in the nucleus, or which is the largest body which is present in the nucleus? It is nucleolus. Nucleolus is the largest body which is present in the nucleus. Okay, nucleolus is the largest body. Now, there are something called as the nuclear matrix. What is this nuclear matrix? You can see here, this is something here. Okay, it is connected with the inner nuclear membrane. The inner one is attached with this nuclear matrix. What is this nuclear matrix? Matrix is anything which gives support. Okay, which gives support. So these are some kind of filaments. These are kind of filaments. We will see what are these filaments. यहाँ पे बस आप याद रखो. These are the nuclear matrix which give structural support to the nucleus. The shape of the nucleus is like this because it has this nuclear matrix. It has this nuclear matrix which is joining these inner membrane with the chromosome, with the nucleolus, with other organelles which are present in this nucleus, so that the shape of your nucleus is intact. If this is not present, the nuclear matrix, if it is not present, your nucleus will collapse. Okay, your nucleus will collapse. It will not be in its perfect shape, perfect position. Why? Because it is something like if the bricks are removed from your rooms, the bricks, they are the building blocks. Okay, the bricks has to be there, otherwise the outer skeleton of your rooms will not be there. The same applies here on this nucleus. These are like the bricks. Okay, these are the bricks which gives the structural support, the structural formation, the structural body to your nucleus. Fine. Now you can see two important bodies here which are called as the PML body and the Kajal body. Very, very important for your 2 marks question. In your B part, it always and mostly it comes that what are these Kajal bodies? The Kajal bodies are present in your nucleus. Okay, the Kajal bodies are present in your nucleus. And you should know one thing. Okay, logically you should remember that anything which is pertaining to the nucleus, anything which is related to the nucleus, it will be doing something with the RNA and the proteins. Just like that. If you don't know actual work of this Kajal body also, and if you see the options in your paper, and if you see something like RNA and proteins and some other things which are not relevant, you can directly mark RNA and proteins. Okay, because why? Because you know that Kajal bodies are present in the nucleus, and nucleus what is the main function of the nucleus? It is the information center for our DNA, for our genetic material. It will be helping to make the mRNA, and this mRNA will be then translated into the proteins. So it is like the information is transferred. The information is transferred. So all these bodies are helping in this. Okay. So the specific function of this Kajal body is that it is an important body which is useful for mRNA processing. mRNA processing. Very, very, very important for your single liners and for your 2 marks, that Kajal body is important for mRNA processing. And what are these PML bodies? We will see. It is pro-myeloleukocyte. We will see what is the function here. It is also related to the transcription. Just one word you have to remember. This is for mRNA processing, and this is for transcription activation. The two bodies which are related to your nucleus, the Kajal body and the PML bodies. Okay, everyone understood what is the nucleus overview? Now see, I'm not going into actually the nucleus, the DNA, the chromatin, the heterochromatin. I'm not doing that because my topic is not the cell organelle nucleus, but I will be focusing on the transport mechanisms, how these RNA proteins are transported and what are the important things which are helping in this transport mechanisms. The nucleus as an organelle, we will be doing it in the some other classes. So have you understood every point here? If any doubt, you can ask me. You understood this? The nuclear pore, the outer and the inner membrane, the nuclear pore, the DNA, the Kajal body, the PML body. Yes? Okay, fine, fine, fine. Yes, fine. Okay, let's move forward. Just what I was explaining you, I have written here. Now, two things you have to remember here is that first thing is that, see, your inner membrane and outer membrane not unique. That means they are not like single. They are not isolated. They are in connections with each and everything. Like your inner membrane is connected with something and your outer membrane is connected with something. We will see the where the inner membrane is connected. But here you have to remember that your the outer membrane of your nucleus is continuous with the endoplasmic reticulum. This also comes for your 2 marks question, that outer membrane is continuous with organelle. The outer membrane of your nucleus is continuous with which organelle? That is your endoplasmic reticulum. And that is why whatever is synthesized in your nucleus can be easily transported to the endoplasmic reticulum, and from endoplasmic reticulum it can go to any part of the cell. Endoplasmic reticulum to Golgi, endoplasmic reticulum to the nuclear membranes, endoplasmic reticulum to the nucleoplasm, anywhere it can go. So the lumen means the space. Okay, the lumen means the space. The space are not isolated, they are connected. It is like a tunnel. आपने देखा होगा ना टनल्स कैसे होते हैं, जो दो पहाड़ों के बीच में से गैप्स होती है, the gaps, and they are connecting it. So the same way, your nuclear and area, your nucleus area and your endoplasmic area is connected with the outer membrane. Okay, with the outer membrane. Also, you can see there are something called as nuclei. I have told you what are these. These are the largest organelle, and their main function is ribosome biogenesis. They will make the ribosomal subunits, 50s, 30s, 40s, 60s, whatever is the ribosome subunit, they will make that, the nucleolus. Okay, the liquid which is present in the nucleus is called as the nucleoplasm. The liquid which is present in the nucleus. Now the liquid which is present in the cell, the liquid which is present in the cell is called as the cytoplasm. Okay, and the liquid which is present in your nucleus is called as the nucleoplasm. Now more majorly now the students get confused here. These types of questions are also asked, that what is the liquid or what is the jelly-like material which is present in the nucleus, and they get confused. It is nothing but it is nucleoplasm. The area which is covered by the nucleus and the liquid which is present in that, it is called as the nucleoplasm. Fine. I have told you about the nuclear pore complex. Now, one thing is there, that what is syncytium? What is syncytium? Syncytium is something which is called as a multinucleate. You just have to remember this word, multinucleate. Multinucleate means there is one cell and there are many nucleus. 1, 2, 3, 4, 5. That means the nucleus are dividing, but the cell is not dividing. Okay, this is something called as the syncytial condition, which is a multinucleate condition, and this is present in your skeletal muscles. Okay, it is present in your skeletal muscles. It is very, very important. If in your 4 marks question, sometimes it is asked that whether the nucleus, whether all the cells are uninucleated. So we get confused. Obviously, we will think that yes, majority of the cells have one nucleus, but no, here is something which is exception. Okay, there are something which have multinucleate conditions, and the multinucleate condition is called as syncytium or syncytial. That is a multinucleate condition, and this also comes in your 2 marks question. the condition in which the cell has a number of nucleus. The question will be like this. Or a multinucleate cell is called as, it is called as a syncytium. Nuclei is different from the nucleus. Yes, who has asked that question? Yes, yes. They are different. Nucleus is a central organ which has the DNA, and nucleolus is present inside the nucleus. Nucleolus is present inside the nucleus. Okay. And it is very important for your RNP synthesis, the ribosome biogenesis. For your ribosome synthesis, it is very important. They are different. Okay, they are different. Nucleus is different and nucleolus is different. And also, nucleolus is the largest organelle present in your nucleus. न्यूक्लियस के अंदर का जो सबसे लार्जेस्ट, आपने देखा अभी न्यूक्लियस के अंदर सब कुछ प्रेजेंट है, डीएनए प्रेजेंट है, काजल बॉडीज प्रेजेंट है, पीएमएल प्रेजेंट है, और भी बहुत सारी चीजें हैं जो प्रेजेंट है, और उसके अंदर से एक चीज जो है वो है न्यूक्लियोलस. तो न्यूक्लियोलस जो है दैट इज द लार्जेस्ट organelle or the largest body which is present in the nucleus. Understood? Clear? Fine? Okay. Okay, Salman. Okay. Okay. So you can see the Kajal bodies, they are involved, they are the bodies which are rich in proteins and RNAs, and they are very important for mRNA processing. I was telling you specifically is for small nuclear RNA proteins. You just have to remember this word, mRNA processing and snRNPs. इसके अलावा कुछ भी याद रखने की आपको जरूरत नहीं है. And what is PML bodies? It is promyelocytic leukemia protein. And ये जो है that is ट्रांसक्रिप्शनल एक्टिव रीजन्स में हेल्पफुल होता है. ट्रांसक्रिप्शनल एक्टिव रीजन्स मतलब कि हेट्रोक्रोमैटिन में कौन सा प्रोटीन जाएगा, यूक्रोमैटिन में कौन सा प्रोटीन जाएगा, वो ये प्रोटीन हेल्प करता है उसको फाइंड आउट करने के लिए कि कौन सा एरिया है जिसको कि ट्रांसक्रिप्शनल एक्टिवेशन में जाना है. सो दीज आर द टू इंपॉर्टेंट बॉडीज विच आर प्रेजेंट इन द nucleus. Okay, these are present in the nucleus. Don't forget this. Okay. Now, this is our nucleus, and this nucleus is telling, as I told you, that this is the information center of your cell. So the information center means it is like a hub, it is like a supercomputer which has all the data which is present here, all the data. So obviously it is a very important, it is having a very important position in our cell. So he is telling that with great powers comes great responsibility. Obviously you can see the bigger positions which are there, the prime minister or the president of the US, or in our family also the father's position, they have great responsibility to perform. So let's see what are these important responsibilities which our nucleus has to perform. Now see, the first is your stability. I have written only the words and I have explained it also, but whatever is depicted here, that is enough to solve the questions. Okay, now just you have to remember this word, that it is important for stability. What is the stability? Stability is that this is my nucleus and this is my cytoplasm. I have written here, this is cytoplasm and this is my nucleoplasm. Now this nuclear envelope or this nucleus is a separate compartment. Now as we grow older, ओके जब हम छोटे बच्चे होते तो इट इज लाइक वी कैन स्टे विथ आवर मम्मी डैडी, उनके रूम्स में वी कैन स्टे, बट एज वी ग्रो ओल्डर वी नीड प्राइवेसी. So we demand that we should have our separate room, like so we can be free, we can do our stuffs, we can do it at our pace with our comfort. The same thing is here. This is a compartmentalization. This is a compartmentalization. Like this is a separate room. The nucleus is a separate room and it is separating it from where? Cytoplasm. So cytoplasm mein whatever is present, it cannot harm our nucleus. And why it is important? Because it contains our DNA. That is why it is very important that we should store it in a very secure place. If we have something very valuable, we ought to store it in our lockers, or we bank lockers like that. So you have to remember it like this, that it has very important thing which is present, which is called as the your DNA, and that is why it is having a separate room. So it is giving stability to your cell. It is giving stability to your cell. Okay. The second thing is your regulation. The second thing is your regulation. Can anybody tell me what is regulation? What is regulation? And one more question, management. Yes, Deepshika, it is kind of management. And one more question from here, stability. In prokaryotes, are the DNA or the chromosome stable? I'm asking a question. In the prokaryotes, whether the stability occurs, whether there is stability, yes or no? No. Why? No. Why? Yes, you are correct. Why? Why? The answer is, what is the reason? Because they have primitive, what, primitive nucleus. Yes, because they lack nucleus. Simple. Because they lack nucleus, they don't have the nucleus. That is why their the full cell is not stable. The nucleus is not, the DNA is not stable. Whatever is the genetic material, it is not called as the DNA. The genetic material, it is not stable. Any harm or any change which is happening in outside will ultimately destroy your genetic material. So your prokaryotic cells are not stable because they don't have this nucleus. Now the regulation means, as Deepshika told, it is called as the management. Okay. Now here the management is what we are managing here. We are managing the genes. We are managing the genes. Which genes have to be transcribed, which genes have not to be transcribed. All these things are regulated here. And how it is possible? Suppose there are some transcription factors which are present in the cytoplasm. Okay. Now what is the function of this transcription factor? What is the function? The transcription factor, as the name suggests, in the last class also I have told you, they help in the transcription. As the name suggests, they help in the transcription. And what is transcription? Transcription is when your DNA information is converted to RNA. When your DNA information is converted to RNA, these factors will help here. Okay, these factors will help here. Now, I will manage that which transcription factor can go and which transcription factor cannot go. So this is a kind of management. This is a kind of regulation which I am doing, that I can allow this one to go, but I will not allow this one to go. So this is only possible when you have a separate nuclear membrane, because these nuclear membranes have something called as the pores, and these pores will decide that which have to go and which don't have to go. Okay, which transcription factors it will allow and which transcription factor it will block. Okay. So this is called as a regulation. Now the third thing is compartmentalization. Now compartmentalization means two things here. You have to know that compartmentalization here I have written in two ways. First is the compartments, that is your outer cell and your inner nucleus. So these are the two compartments, and one compartment is here. Now see, this DNA has been transcribed into mRNA. This DNA has been transcribed into mRNA. These are your nucleotides, A, T, G, C. Okay, these are your nucleotides. Now, what are these things? What are these things? Can anyone tell me what are these things? What are other things which are present in the mRNA? What are the two important things which are present in the mRNA? Phosphate bonds, okay. Uracil, okay. ओके यूरासिल, ओके ओके नाउ सी, हाँ यूरासिल तो है, मतलब मैंने यहाँ पे टी बोला है ना, टी के जगह पे यहाँ पे यू हो जाएगा, ओके वो चीज नहीं है। द mRNA which is present in the nucleus, it is a kind of immature mRNA। ये मैच्योर नहीं है, अभी ये घर के अंदर है। हम घर के अंदर तब तक रहते हैं जब हम immature रहते हैं, you know, when we are small we are not allowed to go outside alone because we are immature, right? But as we grow older, as we get mature, we are definitely, we are going outside and we are learning so many new things and everything. So what are these? These are introns. These are introns. INTRONS. These are introns. And what are these? These are exons. These are exons. And what are the active portions? Exons are the active portions and introns are the inactive portion. So immature mRNA contains both the introns and the exons. And mRNA processing is very important because we need to remove these introns. ये जितने भी introns जो present होते हैं आपके mRNA में, आपको इसको remove करना होता है. That is called as splicing or processing of mRNA. Splicing दूसरा होता है, but एक तरह से आप इसमें बोल सकते हो, ठीक है. So introns जो रहता है उसको हमें remove करना होता है. And जो बच जाता है that is your exons. So सिर्फ और सिर्फ जब exons present होता है आपके mRNA में, that is called as a mature mRNA. Immature mRNA में क्या क्या present होगा आपका? Introns भी present होगा और exons भी present होगा. Okay, your immature mRNA will have these these exons. ये जो आपके circles दिख रहे हैं ये सब exons है, और ये जो आपको lines दिख रहा है ये सब introns है. तो आपको क्या करना है, ये जितनी भी introns होते हैं you have to cut it out before it enters into the cytoplasm. क्योंकि अगर ये इस कंडीशन में अगर ये इधर आ जाता है immature में, तो एक्सीडेंट्स होने के बहुत चांसेस होते हैं. You can understand, अगर कोई छोटा बेबी, अगर कोई छोटा बच्चा अगर बाहर कहीं खो जाता है तो क्या हो सकता है, right? The same thing is here. If this immature mRNA goes to the cytoplasm, there are chances of mutation. There are chances that this mRNA will not be activated. It will not form its protein. Why? Because it is having these introns. So it is very important. It is very important that these introns have to be removed. These introns have to be removed. Okay. And only your mature and processed RNA will come out. Now, if अगर suppose कि हमारे पास ये nuclear membrane present नहीं था, if suppose I was not having this nuclear membrane, I was not having this stuff, तो क्या ये possible होता? ये compartmentalization, was it possible? Was it possible? If this door was not there, if this compartmentalization was not there, what would happen? Directly mRNA banta, in whatever condition it is, directly it would have gone to the cytoplasm, right? And this mostly okay, so that is why your nuclear membrane or this nucleus or this compartmentalization is very, very important. Why? Because a kind of active regulation, tight regulation, strong regulation is important for the transport of this mRNA's protein, for the transport of this mRNA proteins. Okay, so this is your compartmentalization. One more thing, suppose I don't want, suppose this, suppose one signal has come that I need to make some nuclear protein complex, some transmembrane protein complexes. A signal has come. Okay. So what this DNA has got this signal. So this DNA, what will do? It will make its mRNA. It will make its mRNA. But somehow now the signal is coming that we don't want this. We don't want this because that thing has been done from somewhere else. We were having some stock, so we have managed from that. So now we don't want this mRNA. Okay, now the signal is coming that we don't want this mRNA. Now see, अगर ये आपके पास अगर ये वाला मेम्ब्रेन प्रेजेंट नहीं होता तो ये सिग्नल का कोई सुनता ही नहीं. Okay, this would have directly gone and this would have directly started its translation process. But now, since the signal is coming, the signal is coming no. So what this will do, this nuclear pore or the nuclear pore which is present here, it will not allow this mRNA to enter the cytoplasm. And what it will do, even though it is mature, okay, even though it is mature, what I have written here, it is mature and process will come out. But the thing is that it has to get a correct signal. Only then it will come out. If now the signal is coming, no, I don't want this. What will this mRNA will be, what will be the fate of this mRNA? It has to be located here only. It will not come out of this nucleus into the cytoplasm until the signal is okay. Until the signal comes okay. Only at that point, this nucleus can transport from here and it can go to the cytoplasm and it will make its protein. Understood, everyone? These are the 4 important responsibility which the nucleus as a whole, the nucleus, the nuclear membrane, all these perform. Understood, everyone? Any doubt till here? Yes. Okay, Shalini, ask. What is the doubt? Clear? Clear? Shalini was having some doubt. Am I audible to everyone? Hello?
Yes, ma'am, you are audible.
Millie, what question you have asked? एक minute, but how DNA will come to know how to terminate the process of the RNA? How to terminate? No, it will not terminate the process. DNA cannot terminate the process. Okay, termination is something different functioning which is not done by the DNA. DNA can only open up and it will only allow the transcription factors to come and it will only allow the formation of the mRNA. Okay, the only control what DNA can do is that, now see, can you see the shape of this DNA? Okay, can you see the shape? It is close. It is close. Okay, it is like something thread which is crumbled up. So if suppose, just a second now. If suppose I want a gene which is present here to be transcribed, it is located here. Okay. Now there are 2 conditions. If suppose one gene is present here, one gene is present here, and one gene is present here, which is easy to transcribe? Which one is easy to transcribe? Which one is easily accessible? Which one is easily accessible? If this transcription factor is coming and it is locating, yes, obviously Vidushi, right? It is the blue one. Yes, because it is present on the outside. It is present on the periphery. So this transcription factor can easily locate. But if it want to transcribe this gene, the DNA has to open up. The DNA will open. Only in that condition this gene will be targeted and this transcription factor will come here, join, and this can be transcribed into the mRNA. So what this DNA has, the regulation what I was talking here, that is something very different topic, but I'm telling you here in 1 or 2 words that this opening and closing of the DNA is one of the regulation by which it is regulating the transcription of the genes. If it is like this, and one more thing I was telling you, activation, right? Activation I have told you in just a previous slide. The activation is this only. The proteins which will help this DNA to unfold. Only in that condition, this transcription factor will find this. And only in that condition, it will be transcribed. So this is one kind of regulation which it can do. It cannot say no, but it can cover itself. Okay, and how it can cover itself? There are signals. There are various signals which are coming. Okay, there are various signals which are present here. Last class we were talking about cell signaling. Okay, so there are various cell signalings which are coming and which are transporting their messages, what they need and what they don't need. Fine. Mature RNA will accumulate inside the nucleus only. Yes, yes, the mature RNA will, it will be inside the nucleus only, and as soon as when the signal is coming, yes, and all these mRNA will be transported into the cytoplasm, and very quickly the proteins will be formed. In that case, the protein formation will be very quick because the previous work is done already. So some disaster has come into the cell and it needs the protein at a very fast pace. So these mRNA, in that condition, it will be helpful. Okay. Okay, these are the functions which I was just telling you. Okay, now see, this is very, very important. This is your actual structure of your nucleus. This is your outer This is your outer nuclear membrane. This is your inner nuclear membrane. You can see your outer nuclear membrane is continuous with the endoplasmic reticulum. So the lumen, this is called as the lumen. This lumen is continuous with here. This lumen, ये जो आपका space है, perinuclear space इसको बोलते हैं, perinuclear space. You can see here, this is called as a perinuclear space. ये जो बीच का जो light green space है, that is called as a perinuclear space. This perinuclear space is continuous with your endoplasmic reticulum. So if a protein, transmembrane protein which is present here, if a transmembrane protein which is present here, it can be easily transported to the endoplasmic reticulum. And this transmembrane protein, if it has to go to the plasma membrane, it can easily be transported. It can easily be transported. So you should remember that this is outer nuclear membrane, this is inner nuclear membrane, this is your pore. This is your pore called as the nuclear pore. This is called as the nuclear pore. And how this nuclear pore is adjusted here? It is adjusted with a protein called as the pore membrane protein, POM. Okay, it is very, very important. This protein is very important. It is called as the POM protein. That is the pore membrane protein because it is located at the pore. And what it is doing? It is helping all these structure to be holded at this pore. ओके ये जो आपका स्ट्रक्चर है ये इस पोर पे एंकर्ड रहे इसके लिए दिस पोर मेम्ब्रेन प्रोटीन इज प्रेजेंट हियर फाइन so this is your outer nuclear membrane, this is your inner, this is your POM protein, this is your nuclear pore, this is your perinuclear space. One important thing is here which is called as lamina. You can see this pink stuff. This is called as your lamina. Okay, and this lamina is contiguous with which membrane? Can you see it in the figure? It is contiguous with which membrane? Outer nuclear membrane or inner nuclear membrane? Inner one. Yes, very good. So from here there are two types of question. Outer nuclear membrane is contiguous with your endoplasmic reticulum and inner nuclear membrane is in contact with your lamina protein. Inner nuclear membrane is contiguous with your lamina protein. Lamina protein. Lamina is a very important intermediate filament. It is very important intermediate filament and it helps in making, I was talking about the matrix, right? That matrix is the lamina. It will help in making this shape in its correct position, in its correct configuration. This is the lamina protein. Lamina A and lamina B. This comes in your 2 marks question. that which are the proteins which are present in the nucleus for its cell, for its structural organization. It is your lamina proteins. Lamina proteins. Understood? Okay. Now I'm telling you the most important stuffs which are asked in your exam, and which is most probabilities that they will be asked in your exams. Okay. Okay, so this is your nuclear envelope. I have explained you what was the whatever was there in the diagram. One thing is here is the perinuclear cisternae. Perinuclear cisternae is outer membrane, outer nuclear membrane plus inner nuclear membrane plus the space, okay. All these three together, they are called as the perinuclear cisternae. Understood? This is your 2-mark question. This is your 2-mark question. Understood? What is perinuclear cisternae? Outer nuclear membrane plus inner nuclear membrane plus the space. That means all these three. This is outer, this is inner, and the space between them. All the three together, they constitute the perinuclear cisternae. Okay, I was telling here, they are called, the nuclear lamina, they are a kind of intermediate filaments, 30 to 100 nanometer thick, A type and B type. That's it. And one thing is here, they play a very important role in assembly and disassembly during mitosis. Now, what happens in mitosis? The cell has to divide. And what during the cell division, your the nucleus also has to be divided. So at that time, this lamins, they are disassembled. And when the cells are again formed, they are again assembled. Now here, there is a 4-mark question. There is a 4-mark question which I have written here in a note. Now see, if antibodies to lamins are injected into the cell, the nuclei cannot reform. If some kind of experimental condition is given to you, and mostly this type of questions comes in your part C analytical type, that what will happen if anti-lamins are injected into the cell and the cell is dividing? What will happen? What will happen? The cell division can occur, but the nucleus will not form. The nucleus will never be formed. Okay, it cannot reform after the cell division. Therefore, these lamins are very important for what? Assembly of the nucleus. Which is very important for the assembly? That is the lamins. And if you are injecting any anti-lamins, anti-lamin A or anti-lamin B, the nucleus will never reform. Okay, even though the cell is dividing, even though the mitosis has occurred, everything is happening, but the nuclei will never form. So this is a 4-mark question, analytical type, which is asked in your exam. This type of questions is only asked in your exams. Okay, little bit tricky, but the answer is very easy. Now you can see this nuclear pore. I have shown you the pores many times, but you can see actually what is the design of this pore. Okay, there are 8 spokes. Okay, there are 8 spokes present here. 1, 2, 3, 4, 5, 6, 7, 8. Okay, there are 8 spokes. Now you can see here. This is your outer ring and this is your inner ring. This is your outer one. This is your inner one. And it is like pinching off the membrane. And there is something called as a plug present here. And these things, these are like spokes which are coming out. And whatever regulation is there, whatever transport mechanism is there, it is passing through this gap. Through this gap, the transport mechanism happens. Okay, this is your pore complex through which all the transport mechanisms occur. And what is this? There are 8 subunits of the protein which are present, and there is a presence of a central plug, and it is like pinching off the membrane. Pinching off. You can see it is like pinched off. Okay, and here the central plug is present. So this is the structure of your nuclear pore complex, and you can see the spaces here. Through these space only the transfer occurs. Only through these spaces, the transfer will occur. Fine. So this nuclear pore complex is also asked in your exams, and mostly it is asked in your 2 marks questions. Okay, now see. Now some important questions which we have to answer is that how do molecules get through the nuclear pore complex? Now I have told you that there is something nuclear pore complex. Now the question is how the things can pass through this nuclear pore complex, and how do these large polar, now if the molecule which has to be passed is very small and it is non-polar, it can easily diffuse. It can easily diffuse through this. But if it is a large molecule like 50 kilodalton, 100 kilodalton, and if it is a polar molecule like RNAs, then how it will be transferred from the cytoplasm? How it will be transferred from the cytoplasm to the nucleus, or the nucleus to the cytoplasm? We have to answer that question. Now, what targets a protein to the nucleus? If some protein is present here, what will target it? And how the proteins which are built here, like the transcription factors, how they are transported outside? We have to answer these questions, and these are very, very, very important pertaining your exams. Now see what will happen. Okay. Now see, there are 2 proteins, A and B. This is oval, this is diamond shape. Now see, this protein has entered inside the nucleus, but this protein is outside. That means there is something which is present on this that is allowing it to enter into the nucleus. Now, if you see the size of this is smaller, right? But the size of this is bigger. But then also, this has entered the nucleus, but this is remaining outside. Now see again. There are 2 proteins which are present inside the nucleus. Now, only one is coming out, but the other is not coming out. So there is something which is present on these proteins which are targeting this to the specific places, from the cytoplasm to the nucleus and from the nucleus to the cytoplasm. So what are these? We have to answer these questions, and this is very important for your 2 marks, for your 4 marks, both. Now see, suppose I have this cell, and listen very carefully, okay? This is very, very, very important, and if you get this mechanism, everything will be clear, everything. This is your cell, and this is your cytoplasm, this is your nucleus, okay? And this is your nucleoplasm, this is your nuclear pore complex. That means this is your gate. Through this gate, the protein has to enter, or the cargo has to enter. Now what will happen? Now see, this is the protein which has to go inside the nucleus. This is the protein which has been formed in the cytoplasm, and it has to go inside the nucleus. Now, there is something which is called as NLS. It is called as a nuclear localization signal. It is called as a nuclear localization signal. It is something like that. If I give you an example, suppose that there is a lock and there are 10 keys, but the key which is specific to that lock, only that key will open that lock, right? Or if second example, if I give, I know where my home is. I know my address very well. Okay, so why I know my address very well? Because I stay there. So this protein, ये जो प्रोटीन का घर है, the protein houses this nucleus, so it knows it is knowing its address. And what is this address? That is the nuclear localization signal. What is this nuclear localization signal? It is a stretch of sequence of amino acids which is made up of basic amino acids. Very, very important. Lysine, arginine are the are the amino acid which will make this nuclear localization signal. And nuclear localization signal नाम से ही पता चलता है कि it is targeting this protein to the nucleus. Okay, to the nucleus. Now you see, now it knows its address, it knows, but it don't know how to go inside. It don't know how to, it knows that where it is, where is the target. इसको पता है कि मेरा टारगेट क्या है, मुझे न्यूक्लियस के अंदर जाना है, ये इसको पता है, बट इसको ये नहीं पता कि इसको जाना कैसे है. It doesn't know that how it has to go inside the nucleus. So there is a protein which comes to its help, and it is called as the important. It is the important or the karyoferrin. It is called as the important or the karyoferrin protein. And what this protein will do, there is something called as RAN-GTP. There is something called as RAN-GTP. RAN is the small nucleotide protein which helps in the transfer. It is like a crane. वो क्रेन जो कि एक जगह से चीजों को उठा के दूसरे जगह पे रख सकता है, जो भारी भारी चीजों को उठा के रखता है ना, you must have seen the crane. So ये RAN GTP का काम क्या है? It will pull this cargo inside the nucleus. It will pull this cargo inside the nucleus. And what is the name? RAN GTP. Okay, RAN GTP. It is a kind of protein which will help to transfer this into the nucleus. Now how it will do, इतना समझ में आया ना कि there is a protein. This protein has a nuclear localization signal. This nuclear localization signal is made up of 10 to 15 amino acid, which is only made up of lysine and arginine, only with lysine and arginine. And it will bind to the important, because important will help it to enter in this NPC. And how it will help? See, this RAN GTP will come and bind to this important. What was the location of RAN GTP before? What was the location of RAN GTP? It was in the cytoplasm or it was in the nucleoplasm? Not in the inner nuclear membrane. I have just shown in the inner nuclear membrane, but it is in the nucleus. It is inside the nucleus. This question comes in your 2 marks, that what is the location of RAN GTP? Yes, it is inside the nucleus. It is inside the nucleus. And from the nucleus, it is coming and it is binding this important. Now, what will happen? Now, this protein has come inside with the help of this RAN GTP, and this important will go. This important will go because it does not have any work, because it was just a kind of helper protein. It was kind of helper protein which has helped this protein, which has helped this to come to its target place. Something like that. If a small kid has lost his home address, but he knows that what is the address, and some uncle or auntie, they are targeting him to his place. So important is like that uncle and auntie. And nuclear localization signal is the address which you know. Okay, and this RAN GTP will help. It is like a watchman. It is like a watchman. And this watchman knows, yes, this protein is staying in my building. So this RAN GTP keeps a check on which protein is going, which protein is coming outside. So this RAN GTP is like a watchman, and it will carry this protein inside the nucleus. And this important will come out. It will come out with this RAN GTP. Okay, this RAN GTP, with this RAN GTP, this will come out. Now, what will happen to this? What will happen to this important? Can anybody tell me what will happen to this? What you can think? अभी इसका कुछ काम है क्या important का? Degenerate. Okay. Any other answer? Okay. there is a protein called as GTPase activating protein. There is a protein called as GTPase activating protein. Now this GTPase activating protein, it will separate this important and separate this RAN GTP. Okay, now this important is not degenerated. Why? Because this important can carry many proteins inside the nucleus. Okay, so this important is usually not degenerated. It is again coming back into the cytoplasm, and again if any protein is there who is having this nuclear localization signal, it will again bind to it and it will again target that protein into the nucleus. So what will happen is that this gap protein will separate both important and your RAN GTP, and then your RAN GTP with the help of this NTF2, that is nuclear transcription factor, it will go inside the nucleus. Because इसका place क्या है? इसका जो actual place था वो था nucleus के अंदर, वो तो सिर्फ इस protein को लेने के लिए आया था, right? It has only come to take this protein inside the nucleus, so it will again come inside the nucleus. तो जो बाहर रह जाता है वो क्या है? Importance. Importance are outside the nucleus, and what is inside? The protein which was targeted and your RAN GTP. Is it clear, or should I explain it again? Is it clear? This is the important mechanism. This is the mechanism which you have to know and which you have to remember, and I think with the diagram it is very easy. And you should know that important का काम क्या होता है? It is like export and import. Import मतलब क्या होता है कि हम कुछ भी चीज बाहर से मंगाते हैं, right? तो it is like nucleus क्या कर रहा है? बाहर से मंगा रहा है. It is taking something outside. Okay, so it is important will come here. Important. So something is there जो कि न्यूक्लियस से अगर बाहर जाएगा तो भी कुछ ना कुछ प्रोटीन्स आएंगे. वो बहुत इजी हो जाएगा आपको पता करने के लिए, अगर आपको ये वाला पूरा का पूरा प्रोसीजर समझ में आ गया है. तो अगर आपको ये प्रोसीजर पूरा समझ में आ गया है, then your other protein will be very easy. The other mechanism will be very easy. Understood, everyone? This is an example. Can you repeat why RAN GTP does not degenerate? Why should RAN GTP degenerate? Because RAN GTP is like a watchman, ना? उसका तो काम क्या है? It is like it has to guard. It has to guard. अगर वो degenerate हो जाएगा, it is a common protein. Okay, it is not specific for this protein. If suppose this protein name is A, so it is not like that RAN GTP can only recognize A. If there are A, B, C, D, E, F, G proteins which is having this nuclear localization signal, this RAN GTP can recognize all those proteins. So is it logical, or is it is ma'am this is the only pathway? Yes, this is the only pathway by which the things can go in and come out. Only the only pathway. Okay, there is no other pathway. The other pathway is like for small proteins, which I have told, it is the diffusion. But for every bigger protein, you have to pass only through this way only. Understood? Okay, so I was telling that is it useful that we should degenerate RAN? Because जितने बार भी प्रोटीन को टारगेट करना होगा यहाँ पे न्यूक्लियस के लिए, every time RAN has to be made. Every time energy has to be generated. Every time RAN के लिए अलग से time देना पड़ेगा. Is it wise to degenerate RAN? Someone has asked me the question. Please reply. No, right? So understand, Vidushi? So that is why ना तो RAN आपका degenerate होगा, ना तो important आपका degenerate होगा, because they are not specific proteins. Okay, but yes, definitely this nuclear localization signal can be degenerated. Okay, because nuclear localization signal का क्या काम था भाई? उसको तो सिर्फ टारगेट करना था कि तुमको ये प्रोटीन जो है इसका टारगेट न्यूक्लियस है. Otherwise, when the protein is made in the cytoplasm, there are so many organelles. Peroxisome is there, lysosome is there, Golgi body is there, endoplasm nucleocytoplasmic reticulum is there. This protein can get confused that where should I go, or nuclear membrane is there. So that is why this nuclear localization signal is present, which is helping this protein to signal it that you should go inside the nucleus only, and you should not go anywhere else. So after once this protein has come inside this nucleus, this NLS will be degenerated, this NLS will be removed, or otherwise nothing is degenerated. Understood? Okay, so now see here. Now see, I have explained this portion. The just opposite portion, just opposite is when you have to transport anything from inside the nucleus to outside. Just the reverse you have to do, the reverse, nothing else. You just have to do the reverse. What will be the different thing here? यहाँ पे न्यूक्लियर लोकलाइजेशन सिग्नल प्रेजेंट है, यहाँ पे न्यूक्लियर एक्सपोर्ट सिग्नल प्रेजेंट है. क्यों न्यूक्लियर एक्सपोर्ट सिग्नल है? क्योंकि इसको एक्सपोर्ट करना है. इट हैज टू बी एक्सपोर्टेड आउटसाइड. दैट इज व्हाई एक्सपोर्ट सिग्नल इज प्रेजेंट. अंडरस्टूड? एंड यहाँ पर इंपोर्टेंट प्रोटीन होता है जो कि इसको लेकर आते हैं. दिस इज योर इंपोर्टेंट प्रोटीन. यहाँ पर एक्सपोर्टेंट प्रोटीन होता है. देयर इज एक्सपोर्टेंट प्रोटीन. जो भी यहाँ पे मैकेनिज्म हो रहा है ना, सीधा उसका आपको यहाँ पे उल्टा कर देना है, सीधा उल्टा. और इसलिए आपको क्या करना है, एग्जाम में जब आप पढ़ते हो आपको सिर्फ एक ही मैकेनिज्म को अच्छे से पढ़ना है. दोनों पढ़ोगे तो आप कंफ्यूज हो जाओगे. सिर्फ आपको क्या याद रखना है, द इंपॉर्टेंट कीवर्ड्स, द इंपॉर्टेंट कीवर्ड्स जहां पे चीजें चेंज हो रही है. बाकी पूरा का पूरा मैकेनिज्म वैसे ही चल रहा है. बस यहाँ पे न्यूक्लियर लोकलाइजेशन के बदले न्यूक्लियर एक्सपोर्ट सिग्नल आ जाएगा, इंपोर्टेंट के बदले एक्सपोर्टेंट आ जाएगा, क्योंकि इसको बाहर की तरफ जाना है. सी रैंड जीटीपी आपका कहाँ प्रेजेंट है? न्यूक्लियस के अंदर. तो रैंड जीटीपी भी जरूरी है, क्योंकि भाई ये वॉचमैन है, ये गार्ड करेगा. दिस इज अ वॉचमैन, इट हैज टू गार्ड कि क्या अंदर जा रहा है, क्या बाहर आ रहा है, व्हाट इज गोइंग इनसाइड, व्हाट इज कमिंग इनसाइड. without this nothing can happen. So this is a guard protein. So now it has bind here. अब ये क्या करेगा, इसको बाहर छोड़ के आएगा, और जैसे ये इसको बाहर, as NIs have basic amino acid, likewise export protein contain any, हाँ they are mainly they are having hydrophobic amino acid. कुछ ऐसे specific नहीं है उसमें, but mostly they are having hydrophobic amino acids. Okay, mostly hydrophobic amino acid. Nuclear localization signal specific है, क्योंकि वो conserved protein से पूरा बना है, तो वो basically arginine और lysine का होता है. But nuclear export signals are majorly 10 to 15 of continuously hydrophobic amino acids होगा. ठीक है? Then see, this RAN GTP will come and फिर ये बाहर आ जाएगा. अब RAN GTP को फिर से अंदर आना है. RAN GTP has to come inside because उसका जो पोजीशन है वो है, उसका क्या, न्यूक्लियस है उसका घर. तो यहाँ पर क्या होगा, अगेन गैप प्रोटीन विल कम, एंड ये क्या करेगा, इसको डिजेनरेट कर देगा एक्सपोर्टेंट से, और ये जो है ये वापस से इनसाइड आ जाएगा. अंडरस्टूड? अगर एक मैकेनिज्म समझ में आ गया तो इसको समझने में कोई प्रॉब्लम नहीं है. दैट इज व्हाई आई हैव एम्फसाइज्ड ओनली वन. अगर मैं दोनों एकदम ऐसे समझाऊंगी तो बहुत कंफ्यूजन हो जाएगा. आपको सिर्फ ये याद रखना है कि न्यूक्लियर एक्सपोर्ट सिग्नल है, एक्सपोर्ट प्रोटीन है, रैन जीटीपी वैसे ही लगेगा, गैप वैसे ही लगेगा, और ये इसको ब्रेक डाउन करेगा, और ये वापस से इसको अंदर लेके आ जाएगा. फाइन. ओके, सो नाउ वी विल डू द क्वेश्चंस. आई होप आज का आपको सेशन समझ में आया होगा. लास्ट टाइम के क्वेश्चंस में कुछ डाउट्स था, लास्ट टाइम के क्वेश्चंस में कुछ प्रॉब्लम्स था जो सैटरडे को टेस्ट हुआ था. अगर कोई स्पेसिफिक क्वेश्चन है तो आप मुझसे पूछ सकते हो. अगर आपको कोई क्वेश्चन का आंसर नहीं आया हो तो आप मुझसे पूछ सकते हो. ठीक है? नाउ इफ यू हैव अंडरस्टूड you will give the answer here. Now these are some of the questions which have come in your NET exams and in other competitive exams also. What is the full form of NTF? That is nuclear transcription factor. Nuclear transport factor, sorry, not transcription factor, nuclear transport factor, क्योंकि वो ट्रांसपोर्ट करेगा ना न्यूक्लियस में. So it is a nuclear transport factor. Now name the control center of the eukaryotic cell. Very 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 easy. और ये आपके IIT JAM JNU में ऐसा क्वेश्चन आया हुआ है. Nucleus, very good. It is nucleus, nucleus, nucleus. Okay, okay, okay. Which of the following is not a component of the nucleus? Which of the following is not a component of the nucleus? Very good. It is cytoplasm. अब आपको क्लियर होना चाहिए कि साइटोप्लाज्म जो है वो बाहर के पोर्शन में प्रेजेंट होता है, न्यूक्लियोप्लाज्म अंदर के पोर्शन में प्रेजेंट होता है. It is a cytoplasm of the nucleus. It is called as the nucleoplasm. So cytoplasm is not a part of the nucleus. Which of the following microorganism have two nuclei? Which of the following has two nuclei? टू न्यूक्लियाई. ए स्लाइम मोल्ड, साइनोबैक्टीरिया भी किसी ने आंसर बोला. स्लाइम मोल्ड, ए स्लाइम मोल्ड, स्लाइम मोल्ड. Okay, it is paramecium. It is paramecium. Okay, it is paramecium. Paramecium is your ciliate protozoa, which is having two nucleus, a micronucleus and a micronucleus, a macro and a micro. Okay, इसके पास टू न्यूक्लियस प्रेजेंट होता है. एंड मैक्रो न्यूक्लियस में उसका डेली, जो उसको डेली यूजिंग के लिए जो चाहिए होता है ना, डेली यूजिंग के लिए, वो उसके लिए वो इंपॉर्टेंट, वो चीज उसमें स्टोर करके रखता है वो. एंड माइक्रो में वो क्या स्टोर करके रखता है? ओनली द सेक्सुअल रिप्रोडक्शन जीन्स. ओनली द सेक्सुअल रिप्रोडक्शन जीन्स को ही वो माइक्रो में स्टोर करके रखेगा. बाकी जितना भी उसका डे टू डे लाइफ में जो उसको चाहिए होता है, जो भी जीन्स चाहिए होता है, वो मैक्रो में वो स्टोर करके रखेगा. अंडरस्टूड? सो इट इज पैरामीशियम. रेड ब्लड सेल्स आर मल्टीन्यूक्लिएट इन नेचर. ट्रू और फॉल्स? फॉल्स. रेड ब्लड सेल्स की न्यूक्लियस का क्या स्टेटस होता है? व्हाट इज द स्टेटस? यस, यू आर राइट, इट इज फॉल्स. व्हाट इज द स्टेटस ऑफ द न्यूक्लियस ऑफ रेड ब्लड सेल्स? रेड ब्लड सेल्स में न्यूक्लियस प्रेजेंट होता है सिंगल सेल्ड. सिंगल सेल्ड? इज इट सिंगल सेल्ड? रेड ब्लड सेल्स में न्यूक्लियस ही नहीं होता है. द रेड ब्लड सेल्स आर इन्यूक्लिएटेड सेल. इन्यूक्लिएटेड मींस दे डोंट हैव न्यूक्लियस. व्हाई दे डोंट हैव न्यूक्लियस? बिकॉज उसके लिए जो इंपॉर्टेंट प्रोटीन है वो है हीमोग्लोबिन. एंड एक बार हीमोग्लोबिन का बहुत अच्छा से प्रोडक्शन हो जाता है तो जो न्यूक्लियस रहता है वो डीजेनरेट हो जाता है. व्हाई? बिकॉज आरबीसी का मेजर फंक्शन है ऑक्सीजन ट्रांसपोर्ट. ओके, सो उसके पास जितना स्पेस होगा ना, वो उतना ज्यादा ऑक्सीजन को बाइंड कर सकता है. दैट इज व्हाई न्यूक्लियस जो है वो उसका एक्स्ट्रा स्पेस लेगा, इसलिए न्यूक्लियस को डीजेनरेट कर देते हैं. नेम द स्ट्रक्चर विच इज यूज्ड टू ट्रांसफर macromolecules between the cytoplasm and the nucleus. Yes, very good, it is the nuclear pores. ab confuse mat hona. Okay, it is nuclear pores. Name the signal which helps proteins to move in or out of the nucleus. Which is that signal? Which is that signal? Yes, it is nuclear localization signal. Yes, it is nuclear localization signal. Now next, non-membrane bound body of the nucleus which disappears in the later prophase and reappears in the telophase. What is that? Non-membrane bound. ये word से आपको पकड़ लेना है. What is non-membrane bound? Nucleoplasm is actually your cytoplasm. वो non-membrane bound नहीं है. Yes, it is nucleolus. Yes, Charmi, it is nucleolus. मैंने अभी बताया था आपको. Nucleolus क्या है? सबसे लार्जेस्ट बॉडी है एंड वो उसका इंपॉर्टेंट फंक्शन क्या होता है? आरएनए इसके अंदर होता है. एंड न्यूक्लियोलस जो है दैट इज नॉन मेम्ब्रेन. उसमें कोई भी मेम्ब्रेन प्रेजेंट नहीं होता. आपका काजल बॉडी, पीएमएल बॉडी और न्यूक्लियोलस, ऑल दीज आर नॉन मेम्ब्रेन बाउंड बॉडीज. ओके, बट ये जो डिसअपियर और अपीयर होता है वो सारा का सारा जो होता है वो न्यूक्लियोलस में होता है. हेलो, आई एम ऑडिबल? एवरीवन कैन लिसन मी?
Yes, ma'am. Yes, ma'am.
Hello. Okay. Now see, nuclei was first discovered by who has discovered nucleus first time? Yes, it is Robert Brown. Nuclei was first discovered by Robert Brown. Nucleus has, yes, correct, it is Robert Brown. न्यूक्लियस में क्या क्या होता है? सिर्फ डीएनए होता है, सिर्फ डीएनए और प्रोटीन होता है, या सिर्फ डीएनए आरएनए प्रोटीन होता है, या नन ऑफ दीज? Yes, it is very easy. It is DNA, RNA and protein. मैंने अभी आपको रेगुलेशन में बताया कि उसमें डीएनए भी होता है, आरएनए भी होता है एंड प्रोटीन्स भी होता है. Nuclear membrane is in continuous connection with which? Nuclear membrane is in continuous connection with which? Smooth endoplasmic reticulum or rough endoplasmic reticulum? Golgi तो है ही नहीं. Yes, it is rough endoplasmic reticulum. Okay, because इसके साथ जो endoplasmic reticulum present होता है, उसके ऊपर ribosomes present होते हैं and वो protein synthesis में help करता है. So it is rough endoplasmic reticulum. The number of nuclear pore complex depends on which of these? कितना ज्यादा न्यूक्लियर पोर रहेगा किस सेल में, वो किस फैक्टर पे डिपेंड करता है? The number of nuclear pore depends on which of these factors? Size of the cell, अगर साइज बड़ा है तो ज्यादा होगा, साइज छोटा है तो कम होगा, या तो ट्रांसक्रिप्शनल एक्टिविटी पे डिपेंड करता है, या डीएनए कंटेंट पे, या ऑल ऑफ दीज. कुछ लोग बोल रहे हैं ऑल, कुछ लोग बोल रहे हैं ए. ओके, नाउ एक चीज आप मुझे बताओ, न्यूक्लियस का इंपॉर्टेंट फंक्शन क्या है? उसके अंदर क्या है? इंफॉर्मेशन सेंटर है. मैंने आपको क्या बताया था, न्यूक्लियस से कुछ भी क्वेश्चन आएगा, इंफॉर्मेशन है. तो इंफॉर्मेशन इसमें से आपको क्या लग रहा है, इंफॉर्मेशन कौन से वर्ड से डिनोट हो रहा है? इंफॉर्मेशन कौन से वर्ड से डिनोट हो रहा है? जब डीएनए का इंफॉर्मेशन कैसे पास होगा किसी से? डीएनए तो है बट डीएनए कोई, वो तो सिर्फ इंफॉर्मेशन सेंटर है. न्यूक्लियर पोर कॉम्प्लेक्स का काम क्या है? ट्रांसफर करना सिग्नल को ट्रांसफर करना है. डीएनए कंटेंट तो सिर्फ इंफॉर्मेशन सेंटर है. यहाँ पे वर्ड क्या दिया है, व्हाट इज दिस न्यूक्लियर पोर कॉम्प्लेक्स? न्यूक्लियर पोर का डीएनए से कुछ लेना देना है? न्यूक्लियर पोर का डीएनए से कुछ लेना देना है? न्यूक्लियर पोर का काम क्या है? ट्रांसपोर्ट करना काम है ना? न्यूक्लियर पोर का काम क्या है? ट्रांसपोर्ट करना काम है. तो न्यूक्लियर पोर कब ज्यादा होगा? जब ट्रांसक्रिप्शनल एक्टिविटी ज्यादा होगी. ट्रांसक्रिप्शनल एक्टिविटी मतलब क्या होता है? जब आपका डीएनए से एमआरएनए बना हुआ है या कुछ प्रोटीन बना हुआ है, तो जितना ज्यादा एमआरएनए प्रोटीन बनेगा उतना ज्यादा आपके पास न्यूक्लियर पोर्स होगा, ताकि जितना भी एमआरएनए है वो बहुत फास्ट ट्रांसफर हो सके from your nucleus to cytoplasm. Understood? What is the answer now? Tell me. What is the answer? Yes, it is B. Now you have to analyze nuclear pore. ठीक है, डीएनए कंटेंट थोड़ा कंफ्यूजिंग है, बट डीएनए कंटेंट का न्यूक्लियर पोर से कुछ भी लेना देना नहीं है, बिकॉज न्यूक्लियर पोर का काम क्या है? वो ट्रांसफर करता है चीजों को. और इसमें से ट्रांसफर कौन सा वर्ड शो करता है? ट्रांसक्रिप्शन. क्योंकि ट्रांसक्रिप्शन वो है जो डीएनए से एमआरएनए बनाएगा, और एमआरएनए है जिसको ट्रांसफर करना है, या कोई ट्रांसक्रिप्शनल फैक्टर है, जैसे कि न्यूक्लियस ने कोई आरएनपी प्रोटीन बनाया, उसको ट्रांसफर करना है. तो वो डिपेंड करेगा कि आपका ट्रांसक्रिप्शनल एक्टिविटी कितना स्ट्रॉन्ग है, उसके ऊपर डिपेंड करेगा कि न्यूक्लियर पोर कितना होता है. फाइन. Now these are some of the assignment questions. The more the DNA content, the more transcription will occur. क्या humans में, अगर suppose कि आप एक हो और मैं एक हूँ, आप में और मुझ में क्या अलग डीएनए है? आप में और मुझ में क्या अलग डीएनए है? नहीं ना. सिर्फ डिफरेंस कहाँ होगा कि आपके कैसे जीन्स एक्सप्रेस हो रहे हैं और मेरे कैसे जीन्स एक्सप्रेस हो रहे हैं. तो वो जीन्स एक्सप्रेशन के बेसिस पे आपका न्यूक्लियर पोर जो है वो डिफर करेगा. ओके, पोर कॉम्प्लेक्स उस पर डिफर करेगा. अगर सपोज मेरा लिवर बहुत ज्यादा एक्टिव है, तो मेरे लिवर के सेल्स में बहुत ज्यादा न्यूक्लियर पोर कॉम्प्लेक्स होंगे. बट हो सकता है कि, अंडरस्टूड मैम, लास्ट में एक बार सबको आप भी बोलना कि अच्छा. हाँ सर, ठीक है ठीक है. So these are some of the assignment questions. Okay, if you have understood it, all these are mostly single liners. सिर्फ यही एक है जो कि आपको पॉइंट्स में लिखना है. अगर आपको समझ में आया है तो आप ये असाइनमेंट लिख सकते हैं एंड आप मुझे दिखा सकते हैं. मैं नेक्स्ट टाइम इसको वैसे तो मैंने पूरा ही डिस्कस किया हुआ, मैंने इसलिए आपको असाइनमेंट क्वेश्चंस दिए हुए हैं, तो आप इसको लिख सकते हैं और आप मुझे शो कर सकते हैं. फाइन. आप इसको टेलीग्राम पे सबमिट कर दीजिए, आप इसको मेरे मेल पे भेज दीजिए, आप इसको आप टेलीग्राम या मेल पे भेज दीजिए, मैं चेक कर लूंगी. फाइन. अंडरस्टूड? एनी क्वेरीज, एनीथिंग? And if you are liking the lectures and everything, you can join with us. Okay, beta, I'll just check the comments. अभी तक मैंने comments check नहीं किया है. एक मिनट. Ma'am, but how DNA will come? नहीं तो बहुत पुराना. Do nucleus contain proteins? Yes, nucleus contain proteins, ribonucleoproteins. मिली? They contain ribonucleoproteins. जो आपका न्यूक्लियोलस है ना, वो यही काम करता है, राइबोन्यूक्लियोप्रोटीन्स को ही जनरेट करता है, ठीक है? हाँ, ट्रांसपोर्ट होता है साइटोप्लाज्म, आप करेक्ट कह रहे, बट उसके अंदर एक ऑर्गेनेल है न्यूक्लियोलस, आपको याद रखना है न्यूक्लियोलस। तो न्यूक्लियोलस है जिसमें आरएनपी डेवलप होता है, ओनली बॉडी इन द न्यूक्लियस जिसमें कि आरएनपी डेवलप होता है। एंड वही कुछ एक्सेप्शन में भी आता है, वो मैंने यहाँ पे नहीं बताया क्योंकि डेमो क्लासेस में सब पॉसिबल नहीं है, वो जब हम स्ट्रक्चर क्लासेस पढ़ेंगे ना तब हमको और भी अच्छे से पता चलेगा, बट यो द आंसर इज न्यूक्लियोलस। ओके मिली? assignment slide. I think you can take it from the recorded session, Deepshika. Okay, recorded session में से आप answers लिख लेना क्योंकि अभी time हो गया और शायद ये फिर बंद हो जाएगा. Any doubt, anything, any other thing? And if you're liking the lectures, please join with us. Everyone, you can register. Some of the students have registered also. So you can register. Yes, Prema has registered. So there are 2-3 students who have registered and there are more who are asking me the queries to join. So please join with us, okay? So we can start our structured classes and you can get more experience over there. Fine? Any other doubt? Arigdeep sir, if you want to share anything here, you can. Any attendance or anything.
Yes, ma'am. Yes, ma'am. Now, students, now I'm sharing your today feedback link. Please fill the feedback.
So please fill the feedback link, everyone. And you can take the screenshot from the recorded video of this lecture from YouTube, that assignment page, and you can submit your assignments in Telegram or in the ma'am's email. Try to register soon because only a few days left to begin our mainstream class, so try to register it soon. Once again, I would like to repeat, first 30 students will get concession of 500 rupees and you can pay in the installment, just like you can pay 4,000 and then later you can pay 3,500 later. So please register it soon. If you're liking our lectures, just join with us. We will guide you with proper guidance and how to crack and all. Strategy analysis video is there in our YouTube channel. Ma'am has already discussed with students. So once you go through that video, you will come to know how to crack CSIR net program, what are the strategy analysis,